Guys, today's video lecture topic is all about meiosis. As always, please make sure you're filling in your notes organizer as I go through the video. There will be times today where you will need to stop, pause, draw some pictures, write some descriptions, so please make sure to do that. Take the time to do that. Okay, so we've spent some time talking about mitosis, which was the process of creating somatic cells, genetically identical cells. Now we're going to talk about the process of meiosis, which is the process of cell division that is necessary for sexual reproduction. Okay, so quick review about chromosomes and chromosome number. Human body cells, normal body cells, are called somatic cells. They have 46 chromosomes, a set of 23 that you got from your biological mother and a set of 23 that you got from your biological father. Okay, the 23 from your mother was carried in the egg, 23 from father was carried in the sperm. So you have 23 pairs of chromosomes in all of your cells except for in your gametes, which we'll get into in just a second. Okay, so those pairs of chromosomes are called homologous chromosomes. They carry the same genes. Not necessarily the same traits, but they carry the same genes, meaning like they both carry the gene for whether or not you have dimples. But they may not necessarily carry the same, genetic information. They just carry the same gene. Okay, so homologous chromosomes. Okay, so like I said, every cell in your body, your normal body cells, are called somatic cells. They have 46 chromosomes. The only cell in your body that is not a somatic cell are your gametes, which are your sex cells. So in females, we're talking about eggs, and males, we're talking about sperm. And gametes are unique because they only have 23 chromosomes. So think about why that is. Okay, so a cell that contains two sets of chromosomes, or 2N sets of chromosomes, is called a diploid cell. These would be your somatic cells. All of your somatic cells are diploid. They have two sets of chromosomes, two pairs of 23. A cell that only has an N set of chromosomes, or one set of chromosomes, is called a haploid cell. So these are your gametes. Okay, I'm going to use these terms a lot, so make sure you know them. Diploid means 46. Haploid means 23, or half the number of chromosomes. I always think like haploid sounds like half. So haploids have half, one set. Okay, so why in the world then do sex cells need to be so different? Why do they need to have half the number of chromosomes? Okay, so here's some high school sex education for you. The egg and the sperm come together during fertilization, right? So the egg carries 23, the sperm carries 23 chromosomes, and when the mommy and daddy love each other, the eggy and the spermie come together, right, during fertilization, and that number is restored to 46. So an organism produces gametes essentially to maintain the same number of chromosomes from one generation to the next. When the 23 from the egg pairs with the 23 from the sperm during fertilization, that fertilized egg is your very first somatic cell. Okay, that's your very first body cell. So then that fertilized egg is going to start going through mitosis, making more and more cells that have 46 chromosomes, and that, my friends, is how we get a baby. Okay, so now I'm going to go through the steps of, I'm going to go through the steps of meiosis. Please make sure you stop and pause when you need to. Take the time to draw pictures, take the time to write descriptions. Okay, so remember, meiosis is necessary for sexual reproduction. If you have a sexual life cycle, you will utilize the process of meiosis because you have to have a different process to make sex cells. Okay, so the point of meiosis, and I really need to make sure that you understand this, is to make sex cells, is to make gametes. When gametes combine during fertilization, that is when the chromosome number is restored, and then that cell is going to go through mitosis. Okay, so we've got your male and your female. Male produces the sperm, the female produces the egg. These haploid cells, that's produced as a result of meiosis. They join together during fertilization, and that zygote, which is a fertilized egg, is your first diploid somatic cell. And then that goes through mitosis and becomes your either male or female. Okay, so the big picture purpose of meiosis is to reduce the chromosome number in half by separating those homologous chromosomes. So we're starting out with a diploid cell, and we're ending up with haploid cells. So the only way we can do that is by duplicating the DNA once, but dividing the cells twice. So going through two cycles of cell divisions. So those two cycles of cell divisions are called meiosis 1 and meiosis 2. So by starting with a diploid cell and then dividing the cell twice, you're getting haploid daughter cells. Okay, so DNA duplicates once. Here's the big picture. The cell divides twice. So you're going to end up with 4 daughter cells that are genetically different from the original parent cell. So big differences you can see between mitosis and meiosis. And now we're going to get into the details. So here's where you need to pause, stop, draw, and write. Write everything that is happening. Okay, interphase. This is still where the cell spends the majority of its time, even in meiosis, because this is where the cell is growing. This is where the cell is carrying out normal cell functions, maintaining homeostasis. And then the big thing that happens during interphase, we know, is that the DNA replicates. There is only one interphase. We only replicate the DNA one time, but then divide the cell 2 times. That is how we end up with cells that have half the number of chromosomes. So now we're starting meiosis 1, our first set of nucleic divisions, our first division of the nucleus. Okay, so here's just a quick overview, and we're going to go through each of these steps here, but if you'd rather get your information from this slide, that's fine. Okay, so in prophase 1, I want you to put a big old star, and then I want you to highlight, and then I want you to circle whatever you need to do to indicate that prophase 1 is incredibly important, because the big things happen in prophase 1 that do not happen in prophase of mitosis. Okay, so in prophase 1, just like in mitosis, the chromatin condenses into chromosomes, so our chromosomes form. But here's something that they do that is super different. They pair up. They pair up together and they sort of hold hands. Okay, so that forms what's called a tetrad. Tetra, that word part, means 4. So we're actually talking about 4 chromatids, or 2 chromosomes. Okay, so the 2 chromosomes that have been duplicated, the homologous chromosomes, they pair up and they sort of hold hands with each other. Okay, so you can see that happening here. That is very different than what happens in prophase of mitosis. They don't pair up at all, right? They're just sort of floating around. They are pairing up so that they can do what's called crossing over, which I'll get more into in just a minute. But crossing over takes place. The spindle fibers, just like in mitosis, are forming here from the centrioles. The nucleus is breaking down, but big things are happening here in prophase 1 that don't happen in prophase of mitosis. Okay, so they're holding hands. The tetrad has paired up, and they are crossing over. Crossing over is an exchange of genetic information. Think of crossing over almost as if I was wearing a baseball cap and you were wearing a baseball cap, and we took our hats off and we swapped them. So now I'm wearing yours and you're wearing mine. That's sort of what's happening during crossing over. A little genetic segment is literally being exchanged between the 2 homologous chromosomes. This is what creates genetic variation. If it wasn't for crossing over, if you had a sister, like if I have a sister, which I do, me and my sister would look exactly alike. If you had a brother and you are a boy, you and your brother would look exactly alike. But we know that's not the case. We know that everyone has their own set of unique DNA. That is because of crossing over. We reproduce sexually, okay? So we have to have some way of getting genetic variation into our sex cells, and this is how that happens. So crossing over is that exchange of chromosomal segments. So you can see here are your 2 homologous chromosomes. They've colored them, blue and pink to note where the exchange is taking place. So a little piece of the pink has gotten onto the blue, and a little piece of the blue has gotten onto the pink. And so that is going to create genetic variation. That is why organisms who reproduce sexually have genetic variation amongst them, and why asexual reproducing organisms do not. Okay, so we've gone through prophase 1, now we're in metaphase 1. We know that in metaphase, things line up in the middle. So the chromosomes are lining up in the middle. But the difference, and write this in capital letters, in metaphase 1 of meiosis, they are still in pairs. So they are lining up in pairs in metaphase 1 of meiosis. Just like in mitosis, the chromosome centromeres are attaching to the spindle fibers produced by the centrioles, because we know in the next stage they're going to get pulled apart. But the unique thing is that they are lined up in pairs. They're not single file in metaphase 1 of meiosis. So then in anaphase, when the spindle fibers shorten, you have entire chromosomes that are separating and moving to the opposite poles. Remember in mitosis, you had chromatids that were being pulled apart, the little V's. But now we have entire chromosomes because they were just lined up in pairs. So entire chromosomes are separating and moving to the opposite poles during anaphase 1. Take the time, stop on these slides, write down these descriptions, do good pictures. So after they've been pulled apart during anaphase 1, in telophase 1, remember you undo what you did in prophase. So the chromosomes are uncoiling back into chromatin. The nuclei is reforming. Now we have two, though. The cells are beginning to split. The spindle fibers are breaking down. And now some organisms will go through a cytokinesis after meiosis 1, and some organisms do not. So I'm just going to skip that and pretend like in whatever organism we're looking at here, it does not go through a cytokinesis after meiosis 1. Okay, so at this point, we have two haploid daughter cells, but the duplicated DNA has not been separated. Okay, so bear with me. We're going to end up with 4 haploid chromosomes that have just the one set of the chromosomes in them. Okay, so now we have Prophase II. So we've done one set of divisions. Now we're going to go into the second set of divisions, Meiosis II. So in Prophase II, it's very similar to Prophase I. The chromosomes are condensing, or the chromatin is condensing back into chromosomes. The nucleus is breaking down. The spindle fibers are forming. But the difference is that we have 2 cells where this is happening. We don't have crossing over occurring in Prophase II. That only happens in Prophase I. That's why I had you write a big star next to Prophase I, because that stage is so important. Okay, so prophase, actually all the stages of meiosis 2 are almost exactly the same as the stages of mitosis. The only difference is now we're dealing with 2 cells. So in Metaphase II, it's very similar to metaphase of mitosis, just now we have 2 cells. The haploid number of chromosomes are lining up at the equator, but the difference between Metaphase I and Metaphase II is in Metaphase I they were lined up in pairs. In Metaphase II, they're lined up single file, just like they were in metaphase of mitosis. Okay, so they are lined up one behind the other. They are lined up single file. That's a big difference. Underline that, put stars next to it. So after they line up, they pull apart. We know that. That happens in Anaphase II. Anaphase II looks a whole lot like Anaphase of mitosis. Okay, in Anaphase I, you had entire chromosomes that were being pulled to the poles because the chromosomes had been paired up. But since they were single file in Metaphase II, now in Anaphase II, you have chromatids being pulled to the opposite poles, just like in mitosis. The only difference is this is happening inside of 2 cells instead of just 1. Okay, so now we have our little V's, our sister chromatids, being pulled to the opposite poles. So then in telophase, once they reach the poles, the nucleus is going to reform. The chromosomes are going to, uncoil back into chromatin. So take the time, pause on this. And then we're going to have the cutting of the cytoplasm, the cytokinesis. And you still have, remember we learned about the difference between plant and animal cells, the cleavage furrow and the cell plate. You still have that, but it is slightly different for different types of organisms, like different plants do different things. Okay, so cytokinesis, the cutting of the cytoplasm, results in 4 haploid daughter cells, each that have an N number of chromosomes, meaning a 1 set of chromosomes. Now here's something interesting. In males, we produce 4 sperm, each with a set of 23. And each one of these sperms could fertilize an egg and, produce a baby. But in meiosis in females, for some reason, and we're not really sure why, the majority of the organelles and cytoplasms sort of push to 1 cell, and the other 3 are what we call polar bodies. They don't actually mature into eggs. Only 1 actually matures into an egg. And we know that this even happens during the development in the womb. We're not doing this throughout our whole life, unlike males who produce sperm throughout their entire lives. So from 1 meiosis, we produce 4 cells, 4 cells in females, but actually 1 only matures into an egg. Just a little interesting fact there. Okay, so again, let's review. The importance of meiosis: it produces 4 haploid daughter cells that are not identical, which results in genetic variation. Remember, we have that crossing over, that exchange of genetic segments that occurs that produces genetic variation. This is the point of meiosis. I need you to make sure that you know this, is to produce gametes. The point of meiosis is to make sex cells. Sex cells are unique, so they have to have a whole separate process to make them. Okay. So depending on how the chromosomes line up along the equator, okay, remember they line up in pairs and then they line up single file. You have 4 gametes that have 4 different combinations of chromosomes that can result. So depending on how they line up, and then we have the exchange of genetic information during crossing over, those 2 things are how we are different as sexually reproducing organisms. That's what produces genetic variation. That's really important because for sexually reproducing organisms, there are benefits to being genetically varied. So your last quick review review, and hopefully you know this by now, but mitosis, which is a form of asexual reproduction, you have one parent producing genetically identical offspring. Meiosis, which is necessary for sexual reproduction, you have two parents, and that produces genetic variation. And last, here's a slide bringing a mitosis or meiosis related joke for a little extra bonus point. Have a great day!